Alright, so here is my poster of nerve communication and muscle contraction. So we'll start with the stimulation of the dendrites here. I'll go into depth about how the action potential and the signal travels through the neuron and then out the axon terminals to the muscle. And then I will talk about the release of acetylcholine at the neuromuscular junction. I also will go through the muscle contractions here and the two theories, the excitation contraction coupling theory and the sliding filament theory. And then we'll kind of go back to the end where the muscle is relaxed and the acetylcholine is released from the neuromuscular junction. So basically you have a signal from a different neuron that connects to our neuron here, our big neuron here at this, which makes a synapse, which is a connection between neurons. So the dendrites connect to them, and the signal will be passed on through the cell body, along the axon, through the axon hillock. Graded potential will be in here, and I'll talk about that a little bit later, and it'll be sent on and out. But basically, so how we do that. So zooming in on the types of synapses. The main one I'm going to focus on is the chemical one, but there's also an electrical synapse as well. So the action potential will move down, and the depolarization of the neuron, opening calcium voltage gated channel. So the action potential comes down, and it opens up these voltage gated channels. So the calcium will go into the neuron, and it will dock on these proteins. And that allows the membranes to move down to the end, closer to the dendrite and the synapse, the synaptic area here. And it will release the neurotransmitters so that they can dock to a chemical gated channel that's on the receiving neuron on the dendrite there. So there's your chemical gated channels on the dendrite, your neurotransmitters, and all of that is passed on in a chemical synapse. Your electrical synapse is similar, but yet these are the two neurons are already docked, and the action potential can come down. And basically, if you have an action potential in the first neuron, you'll have an action potential in the next neuron. But it's super fast signals, but it's super hard to control. So there's the downfall of an electrical synapse. So talking about repolarization and depolarization. So basically that means that on your dendrite here, let's say on here, sodium will go into the cell, the dendrite, which is the process of depolarization. So it's a trigger of an action potential. So when sodium goes in, you have depolarization. When you have repolarization, it means potassium leaves the cell. So the potassium comes out, and it goes into a synaptic area or outside of another cell. And it's a process of stopping in the action potential. In order to have equilibrium inside the cell and outside, inside and outside of the cell, you have to have sodium-potassium pump. So there's a sodium-potassium pump in here as well, and here as well. But in order for it to have equilibrium, the sodium-potassium pump pumps out 3 sodium for every 2 potassium that come in and out of the cell. So, and it does that by ATP triggering it, and ADP as well, with its inorganic phosphate. So we'll move on to talking about an actual action potential and the graphing of it. So once again, action potential, rapid depolarization and repolarization of a neuron. So when the sodium voltage gated ions approach negative 55 millivolts, it will open the sodium voltage gated ion, allowing sodium to go into the cell. So right here, you can have, you're below negative 55, but once it hits 55, those channels open and you have depolarization, so spikes. Eventually, the cell will move away from the negative 55 millivolts at the voltage-gated ion, and the voltage-gated ion will close. So once that occurs, it moves away from the negative 55. You hit 30, positive 30 millivolts, and you repolarize. So there goes your potassium and sodium. So sodium in and potassium out, just like that. You have two other types as well. You have your, let me back up. You have your graded potential as well down here, right when it hits negative 55. So that would be in here on the neuron at this point. When you start to depolarize, the potassium voltage-gated ions open when you hit positive 30 millivolts. So that allows for potassium to leave the cell, repolarization. And in definition of what a graded potential is, it's the movement of threshold toward or away negative 55 millivolts. Temporal summation. It converts a rapid series of weak pulses from a single source. And a good way to notice that difference is just small little waves before you hit the negative 55 millivolts here. And then spatial summation is several weak signals from different locations and are converted into a single large one. And that one has kind of these higher bumps and waves as well. Okay, so we talked about the actual potential up close and what's releasing and all of that, and the sort of potassium pump. So we'll go on to how the signal actually goes through the whole cell. It's pretty simple. It's just rapid depolarization constantly. So it's depolarization of a segment, of the axon, which triggers depolarization of the next one, and it just keeps repeating by potassium out and sodium in constantly, making the signal move down and through the neuron. So it'll go out all the way out. Just constant depolarization. So step 3, now looking at the acetylcholine release at the neuromuscular junction right here. So, and also we'll talk about the clearing of the neuromuscular, or the acetylcholine. So, just in general, so the neuromuscular junction, meaning the junction or space between a neuron and a muscle. The neuron generates the action potential, which sends it to the muscle. The neuron will pass on the action potential to the muscle by releasing neurotransmitters, which can be inhibitory or excitatory. So this is a very zoomed in picture of what's going on down here, and mainly down here. So outside of the cell, there is a high concentration of sodium and calcium, and both want to go inside the cell, but are blocked by the voltage-gated channels. Within the cell is an excitatory vesicle for muscle contraction that contain acetylcholine. So here's your acetylcholine right here, inside the cell. At rest, inside this whole neuron, it's negative. But once the action potential comes down, the neuron, it will change the inside of the cell to become more positive. And that change in charge will open up the calcium-gated channels, allowing calcium to come in and in from the outside, and then that makes everything else more positive, causing the vesicles that contain the acetylcholine to come down here and release the acetylcholine to go into the synaptic cleft. And then the acetylcholine binds to the sodium channels. So these are the sodium channels here on the muscle. And that causes the channels to open, and a huge influx of sodium ions go inside of the muscle fibers. So action potential, change in charge, acetylcholine comes down, releases it, comes onto the sodium, and then goes inside the muscle. Then the sodium inside the muscle will generate an action potential within the muscle fiber, causing muscle contraction. The sodium coming in creates that action potential, and your muscle contracts. After contraction, the sodium leaves the muscle out the sodium channels, where acetylcholinesterase breaks down the acetylcholine into acetate and choline. That process right there, where the acetylcholinesterase breaks down the acetylcholine, is the clearing of the acetylcholine from the muscle. So technically that's the end of the entire thing, but we'll move on to more of what kind of happens inside of the muscle. So we'll start with the excitation-contraction coupling. So it's the sequence of events by which transmission of an action potential along the sarcolemma leads to sliding of myofilaments. So step one, the events at a neuromuscular junction set the stage for coupling excitation contraction by providing excitation. So step 2, right here, where they release into the muscle. So the release of acetylcholine binds to the receptor proteins on the sarcolemma, and that triggers the action potential. So the action potential travels along here to the T tubules, and it travels down the T tubules where there's these calcium carrier channels that holds a bunch of calcium. So the action potential traveling along the T tubules causes the gates to open, allowing calcium to flow into the cytosol. Calcium then binds to troponin and removes the blocking action of tropomyosin. When calcium binds, troponin changes shape, exposing binding sites for the myosin on the thin filaments. So a little bit closer look. I don't know if you can see it very well, but I have one myosin in the myosin head, and you have calcium that's binded onto the troponin C and troponin T, and the myosin heads actually bind with ADP, and they pull, and that would be the contraction or the power stroke. So, in other words, myosin binding to actin forms cross bridges and contraction begins. Then the excitation theory is over. So here's your sarcolemma. Your sarcoplasmic reticulum would be what the calcium's being held in. Your dehydropidine, that word, receptors are outside over here, and then the ryanodine receptors are also really close to the diphenhydrogen receptors. So yeah, there's your excitation contraction coupling, and up here is just your sliding filament theory. So you have one strand of myosin or muscle fiber, and this is the most popular theory concerning muscle contraction. It states that muscle contraction involves movement of thin filaments past the thick filaments. Sliding continues until overlapping of the thin and thick filaments is complete. So the distance between the Z line decreases here, the I bands shorten right here, and the A bands move closer together. So just contracting. Now, there you go. There's your whole in-depth process of a muscle contraction traveling through the neuron at the neuromuscular junction, the release of the acetylcholine, and release of calcium to the troponin Cs and the tropomyosin and the myosin head pulling. Okay, well, hope you like it. Wasn't for my girlfriend, I wouldn't have these awesome pictures. Thank you.